I think it should be recording. Let me see. All right. All right. So let's go ahead and begin. So this is 2.02, right? So we're going to be talking about microscopes today. All right. So over here, right? We're talking about cells, right? So microscopes and cells. We're going to talk about all those kinds of things, right? So it says all living organisms are composed of cells, right? And so how small are cells? So this picture over here is kind of giving you some examples, right? So the picture on the left, right, all the way to the left, you're just looking at the leaf, right? You're just looking at the leaf, right? There's no magnification being used, right? Now, when you multiply it 10x in this plant, then you start to see the individual cells, right? You see all those little dots; those are kind of like the nucleus of all of those cells. And if you zoom in 60 times, then do you see all of these boxes, right?
Yes.
All of those boxes, right? Then each of those is a cell. So this just kind of give you like a idea about like how small cells are, right? If you zoom in about 60 times, then this is what you're going to see, all right? So knowledge and technology. So basically, the idea here is that they're saying that a lot of times, right, in order to advance in one field, right, you have to first advance in another field, right? So if you say, look, I want to study more and more biology, or I want to study more biology, sometimes that's limited by how much engineering and physics and other things have progressed, right? Why? Because you need those other subjects in order to build tools so that you can study biology, right? Right. So it's
Hmm.
not like a biologist did not, like it's not biologists usually who are inventing microscopes and advancing microscope technology, right? Because to
Hmm.
do that, right, you need like physics, or you need engineering, you need all those other kinds of things, right? You have to build better lenses, right? So that's not biology, right? You have to advance in other fields first, and then when you advance in one field, then you advance in another field. Does that kind of make sense? Right.
Hmm.
So that's just kind of what it's that's kind of what it's saying. So for example, like if like if we want to go to space and study like Mars, right? So most of that work is not done by like astronomers. It's not done by astrophysicists, right? Most of that is done by engineers who have never studied astronomy, right? They
Hmm.
build new kinds of equipment. They build robots, and okay, how does this thing survive in this in this kind of conditions? So it's mostly engineers doing all those work. But then, in order for astronomy to advance, you have to first do the engineering first, right? So that's just kind of what this is saying, right? So let's take a look at some early microscopes. So this picture right here, is kind of showing you what is believed to be like the first kind of microscope, right? So you see this guy von Leeuwenhoek, right? I don't know if he was from like the Netherlands or something like that. So this is basically like the first microscope that is used, right? So you can see on this picture, right? There's kind of like a tiny lens, right? There's that tiny hole which has like a tiny lens, right? And then
Hmm.
on the edge of that pin, where it says sample holder, you put like a drop of something, or you attach like a solid piece of something, right? And then you look through it from the other side, right? Now in this case, the light, right, comes from where? Where do you think the light comes from in order to use this kind of microscope? All right, Freya, what do you think? Probably from the sun, right? Right. Also, I think Sophia, the camera should always be on. So those are just the rules if they come in, right? So the what you're gonna do, right? So let's take a look at the next picture, right? This is how you kind of use this microscope. You take a look at this, right? So you put
Hmm.
the sample on this pin, on the edge of this pin, right? There's a lens, and then you're looking at it, and the light comes in from the sun. All right? Does that make sense? Right?
Yeah.
So this is basically this person Leeuwenhoek, right? He made some drawings, right? So he put in like different kinds of like pond water and all that kind of stuff, and he found all these tiny little things floating around that you couldn't see before, right? So these are some examples of some of the things that he drew, right? So some of them were not necessarily like microscopic. He just magnified them and saw them, right? But some of them were actually things that he called like this animalcules, because they were like moving around and stuff like that, right? So these are like very tiny microorganisms that he found in like pond water or ocean water or whatever he was looking at, and he saw them using this microscope. So these are some more examples right over here of like some of his drawings, right? So these are some of the early drawings, right? Right. So how do you think this microscope is different, right? Can anyone tell me like how this is called the Hooke microscope, right? So this was invented like a lot like much
And
later.
wasn't this guy the guy that named cells?
Yeah, so we're gonna talk about that in a second, right? So how what's different about this one? Can anyone tell me like what's kind of different? Fariha, what do you
Brother,
think is
this
different about this one? Hmm? Which thing? Sparrow? Small thing. Okay. What do you think?
brother, I think this thing has more lenses, brother.
Mhm. Okay, good. All right. So Yusuf, what do you think?
I think this thing has more lenses
More
than the
lenses. Hmm.
yeah.
And Anas, what do you think?
This has like a better focus.
Hmm. So this is just overall the construction is probably better, right? So one of the things you said, there's more lenses, right? So this one
Mhm.
probably has at least two lenses, one in the eyepiece and one in the objective, right?
Mhm.
In this case, right, what is it using for light, right? Its source of light is a little bit different here.
He's using the barrel, brother.
He's using like an oil lamp, right? So he has a lamp and he's going to focus the light through the water flask and in order to shine it on the specimen.
Oh, that's smart,
You
brother.
see that, right? So in this case, this is a little bit different, right? And so in this case, this person, so this is like a more advanced kind of microscope that was used, right? Now, what is this
Mm.
showing? Anyone ever seen this picture before?
These are cells, right?
These are cells, right? So this is where the idea of cells came from. So he took a look at this specimen, all right? So this is from cork, right? You guys know like the cork tree, right? So the tree on the
Mm.
bark, I think like, like the cork, right? Like people make cork board where they put stuff, right?
Yeah.
That's like the bark of this tree. So it's very soft, and you can put pins in it and holes and stuff like that, right? So that's where
All right.
cork comes from. So these are some cells of the cork, right? Does anyone know like why they're called cells? Anas, like look at it, like why did he call them cells?
Because they
I just say
yeah,
so.
I don't
Anas,
They're
know the
really
answer, but I'm not sure.
small
they're very
and they
small.
make up like a whole tree.
And Anas, Yusuf, what do you think?
Because they're like in these little empty spaces of the plant, so it looks like cells,
Right.
like room cells.
Yeah, like prison cells or something like that, right?
Yeah,
So
yeah.
that's he looked at these and thought, okay, these look like prison cells or something. So that's why he named them cells, right? So each of these is like an individual unit. So this is like one of the original photographs. I mean, what pictures that he drew, right? So the things that we've been looking at, these are light microscopes, right? These are like typically, this is like what we think about when we think of a microscope is a light microscope, right? So there's different kinds of light microscopes, all right? So this one right here is called a compound light microscope, right? This is like the typical one that you see, right? So how does it work, right? So you put like a specimen on here, right? You have like a thin section, right? It can't be like thick, right? Because light has to pass through it. So you at the
Yeah.
bottom here, you have like a light, or you have a mirror. Sometimes they have a mirror, and then you use the light from the sun and
Yeah, brother, I
angle it
have one
up.
of these, and it's like a diamond mirror thing,
Hmm.
like at the
Yeah,
bottom.
something like that, right? So you have like a mirror.
Mhm.
You can have a light bulb, whatever it is, and so the light shines through the specimen, right? You're not shining it on top of the specimen to look at it. It shines through the specimen, right? So in this case, do you think that the specimen has to be thick or thin? Freeha, what do you think?
Thin.
Freeha, make sure this is unmuted. Right. So do you think the specimen should be thick or thin for the light to go through? Thin, right? So they call them thin sections. You have to try to make it as thin as possible, right? And so then the light travels up through a series of lenses
What
and
if it's like
up to the
blood?
eyepiece. Right. So well,
If you're trying to see like a blood cell,
yeah. So
doesn't
good.
it look really thick?
Right. So that's called a blood smear. So you have thick smears and thin smears. So you have to try to make it as thin as possible. You have to try to spread the blood out as thin as possible, right? So if it's a liquid,
Okay.
it usually kind of spreads out on its own. But you try to, if it not, you kind of spread it as much as possible.
So you have to like smudge it.
Yes, yeah, basically, right? You kind of spread it, not smudge it, but you spread it, yeah. All right. So this right here on the right is kind of giving you like a real picture, right? So on the bottom right, you have these objective lenses, right? Right. So what is the purpose of these objective lenses on us? Why do we have like these different kinds of lenses over here?
Different zoom levels,
Different
probably.
zoom levels, right? Different magnifications, usually from 10 to 100.
40 to 100, really?
To 100, right? So 10 to 100 down here, I think even less than that sometimes, right?
No, I have a 40 one that goes up to 40.
So it depends. Some of them go up to 100. Now,
Oh,
what you do is at the top there's another lens. Remember, there's an objective lens down here, and then at the top you have ocular lens where you look at it, right? And usually the ocular is about 10. So what you do is you multiply to get the total. So let's suppose the objective is 40,
10 times 10, like 100.
right? So let's say the objective is 40, like in Yusuf's, and the ocular is 10. So what total magnification do you have?
4.
50.
400. You multiply.
You multiply it?
The 40
Oh, you don't add it?
times 10. So that is so if your eyepiece is probably 10, whatever it says on there, probably 10x or 15x, you
Mm.
multiply that times that 40, right? So whatever
Oh.
is your objective magnification times the ocular lens, you multiply them and you get your total whatever you're magnifying.
Oh, right, yeah. My one has the magnification on the ocular lens too.
On the
so it's
yeah, there's probably a number on there, right? Now over here you have something called coarse adjustment and fine adjustment, right? So Fariha, what do you think is the difference? It says over here coarse adjustment and fine adjustment. What do you think that's for? No idea. So Yusuf, what do you think? Just you guys can take a guess. What do you think?
To like adjust like what angle. You see like there's two parts.
So,
Like
so
the course adjustment probably like adjusts like the bottom part
mhm. So we're basically
and the font.
so the idea is to like like if you look at a lens, right? The thing has to be the right distance to be in focus, right? So this is moving the stage, right? You see this where you're holding the slide. This is called the stage, and so you're moving it up and down, up and down. So the course moves it up quickly, but when you get close, then you use a fine adjustment to go like really to
Oh,
you
so
know,
basically there's like less cycle speed, the fine adjustment.
right. So the
from the
fine
coarse
adjustment
one.
moves the stage up like really slowly. The course moves it up quickly, right? So once you put the slide on, you just move it up quickly first, just to kind of get it close. But once you're close, then you use the fine to get it like exact, so that it's really in focus and clear. Otherwise, the whole picture is going to be blurry, right?
Mm.
Let's go over here. So there's different kinds of microscopes that use light, right? So the one that we just looked at is called compound microscope. That's where you put a slide on it and the light comes through it, right? This is called a dissecting microscope. What's like the difference, Anas? What's like the difference? What is different between this one and the one that we just looked at?
The light microscope, you probably get like a better view, and this like just breaks everything apart.
Mhm.
Probably.
Yusuf, what do you think?
Dissecting microscopes are weaker than compound light microscopes are like less magnification.
Usually they're weaker, right? Fariha, what do you think?
Mhm.
Right, what's like the difference between this microscope and that one with objective lenses that we saw?
There's no objective lens in this.
Right, so there's no objective lenses, right? So it says dissecting, right? So in this one, you don't usually use slides, right? In this one, right, you're actually shining the light from the top, and then you're looking at the surface of something. So in this one, you're not using slides, right? This is like, let's suppose you put like a frog on it, and you're dissecting, right? Does the light pass through the whole frog?
No.
No, you're shining it from the top. It's like if
Oh,
if
from
I'm
the top.
looking at something, I shine a light from the top, and I use like a magnifying lens to like zoom in, right? So in this one, this is not you can use this for slides if you have like a light at the bottom. Some of them have a light at the bottom also, but usually this shines light from the top, and the light goes down and bounces off of the surface so you can see it. So this is not for like a thin
So like a
section.
mirror-ish.
Kind of like a mirror, right? So let's take a look, right? You
Or
see
there's probably like a sheet of glass which just spreads the light.
kind of. So look at this picture, right? You see this picture? There's no slide here, right? This is an ant, right?
Okay.
Let me ask you. In this case, does the light pass through the ant, or you're shining it on the top and then
It's
it's bouncing
shining
off?
on top
On
of
the-
each other. Shining on top of it.
On the top? Freya, you understand the difference, right? In one, right, regular with the slides, you're passing the light through the bottom. In this one, you're like shining the light on the top, of the ant and then looking at it, right? You understand the difference, right? So this right here, right, is a dissecting microscope. So that's what they call it, right? Because you're like dissecting something, right? It's not for slides usually. And then here, so some of them they even have like this camera on the top, right? You see this camera, right?
Yeah.
All right. So we have the camera. So we actually have one of these, but it's like here at the school, but it's like a low power one. It's not like a super high power one, but it's still pretty good though. So this is giving you like the different example, right? So Anas, can you identify for me which one came from the compound microscope where you shine the light through the bottom, and which one comes from
Number
the
one.
dissecting one? Which one comes from the dissecting where you shine? So tell me which one is which.
The first one's dissecting. The second one's light.
Right? Yusuf, do you agree?
No, brother.
All right, what do you think?
Oh wait, no. He said this. The first one is the dissecting one, right? Or the
Yeah,
the
that's
compound.
Yeah.
Okay, yeah. Then I agree. If the first one, if he said the first one is dissecting, then I agree.
Right. So you notice the second one, right? Fariha is obviously the slide, right? Right. The second one over here on the right. This is basically like the slide, right? The one on the left, this is like some solid object that they put on top, and now they're looking at it from the top, right? So they're shining some light on it, and they're taking a look at it. So it's the same thing, right? This is like a plant ovary or something like that, and so they're looking at it through a thin section, and then just looking at it, right? All right. So this is basically the same thing. All right. This is the compound light microscope with the objectives, right? So in this case, remember, usually they go up to 400 or even 1,000 times, right? So this is basically about the maximum kind of magnification that you can use with the light microscope. Beyond about 1,000, you can extend it. I think usually some people they try to extend it to like 1,200, stuff like that. But really beyond that, light is not really useful. You cannot use a light microscope beyond that. You can kind of expand the what do you call it? The I mean, you could like make the picture bigger, but you're not going to have the detail. It's not like it's like if you take a picture that has low resolution, right? If you try to zoom in, right, are you going to see like a whole lot of detail?
No.
No, right? So it's the same thing. The compound light microscope, beyond 1,000, you could zoom in more, but it's just going to be blurry. You're not going to see like a whole lot.
And it's gonna be like pixels, less pixels.
Right. It's kind of going to be kind of like pixels, right? Right. So, in this case, right? So okay. So these are some examples, right? So these are just some examples of like some slides, right? Now notice on the left here, this one on the left, okay, this is from a plant, right? They like took a stem and then they cut through it. All right. So is this what it really looks like? Freeha, what do you think? If you just immediately cut it, bang it into a section and put it on a slide and look at it in the microscope, is it going to look like this on the left, or is there something different about it?
Sorry, can you ask the question again?
So this one on the left, right? Is this how a plant just looks like? Like, let's suppose I take a plant, I just cut it, and I make a thin section out of it, and I put it on a slide and look at it under the microscope. Is this what it's going to look like, or did they do something to it first?
They did something
Just
to
like-
it.
They do something to it first.
What did they do to it first?
Add a type of liquid.
They added something, right? So Yusuf, what do you think they added?
They had a dye or a color or something like
Some
that.
kind of color, right? So these are all these things here are stained. These are all called stains, right? So what
Mhm.
they do, just like you stain a T-shirt or anything like that, there's there are certain dyes that you put on the thing, and then you wash it off, right? And so when you wash it off, certain structures take up certain colors,
Mm-hmm.
and so
So
the
it
purpose-
helps to see them, right?
right. So it just helps you see them. So if you don't do that, most of this is just going to be clear. It's just you're going to kind of see it, but you're going to barely be able to see it. Okay.
If you put a color like a certain color,
We can see like the cell
Wait,
membranes
just a second, Anas,
too.
just a second.
be that color, then how would it be different?
Oh, sorry, can you repeat that for you?
Saying if you put a color and like wouldn't all of that like that's the plant would be that color.
So here, good. So here's what happens, right? So they put the dye on it, right? So you have a slide, you squeeze the drops of the dye on it, right, and you let it sit there, okay. However, after that, you let it sit there for like whatever it is, different dyes at different times. Then you wash it off. You wash it off. So when you wash it off, most of the dye goes away, right? Except for the certain structures take it up. Okay, certain structures take up the dye, so the dye is not going to be present all over the place anymore because you washed most of it off, right? But certain parts of the plant or the blood or whatever take up certain colors, right? So when you wash it off, that's why the whole thing is not like red, because you've washed most of it away. Does that kind of make sense?
Mm-hmm.
All right. So these are all different kinds of things. Mm-hmm.
I have a DBA at 945.
Okay, no problem.
Book 940.
Okay, no problem. All right. So so Anas, what do you think this is over here in the bottom right? What is this here?
Bottom right, it's like a photo taken with like a light.
Mm-hmm. But well, obviously it's a photo taken, right? But what is it? Like, what is the thing?
It's
Any guesses?
it's like cells.
Cells, right? Yusuf, what do you think that is?
I think they're cells, brother.
What kind of cells?
No,
What-
it's probably something inside the cell
Oh, what
membrane.
kind of cells?
yeah. What kind of cells? Where is this coming from? Freeha, do you
They're
think you have
transparent
these?
cells.
This one over here on the bottom right. Anyone can guess like what kind of cells these are? Where is this from?
Plant cells.
Plant cells.
Blood cell.
Brother, they're just
Blood
really
cells, okay.
transparent, like
All right, so she got it. Freeha got it. Blood cells.
because they're circles.
Yes,
Aren't blood
right?
cells like reddish?
Yeah, so these
But
are all red. Right?
They're
Those are all red.
literally red. Are you?
Right. All right, Anas
Where
is
they're
red.
pink.
All right.
Honest, they're maybe
Alright.
they're not like perfectly red. Is that an
Alright.
issue?
All right,
Oh,
so,
good,
all right,
bro.
so this is red enough, right?
Okay.
So they're red, and these larger ones. What do you think these larger ones are? These are the red blood cells. What are these other bigger cells?
Blood like clots. Or
Those are the white.
something,
Those are the
or
white.
probably
Those,
like viruses.
those are the white blood cells, right? So
Oh, white blood cells are bigger than red blood cells, brother?
yeah. So red blood cells they carry the oxygen. White blood cells they like fight infections and stuff like that, right?
But they're not even white, brother.
No, but
Like,
remember,
Am I
remember,
colorblind or something?
Oh yeah, yeah, they're dyed. They're
remember they're
dyed.
stained, right? So these ones pick
Yes,
up certain
they're
stains,
stained.
right? All right, good. All right. So this is basically the same thing. This is just showing you. So there's like videos that show you like what kind of staining that they use, right? So you put on different dyes, wash it off, put on other dyes, wash it off, and so that helps you see certain cells better, right? So now, let's suppose we take this picture on the left, right? So I take this picture on the left and I zoom in more, right? So I zoom in more, but magnify, and it becomes bigger. But if it's blurry, right, it doesn't matter all that much, right? So there's two things that when you talk about microscopes, there's two things that are important. It's not just the magnification; it's also the resolution. Resolution, right, is like the detail that you can see. So there's an important concept, right, apart from magnification, called resolving power, right? So in resolving power, let me see if I have a picture here. In resolving power, what is resolving power? Resolving power is the ability to see two objects separately. Okay. Now, so if you magnify on something, right? Let's say there's two objects that are really close together.
Hey, brother, isn't resolving power the clarity of the object?
Right. So that's what
Yeah,
I'm saying,
it's
right?
the resolution.
Right. That's what I'm saying. So here's how you define it, right? It's like if you have two objects that are really close together, right? Is your microscope able to tell that those are two separate things and not one thing blurred together? You understand, right?
Oh, all
So
right.
so same thing with like telescopes, right? Let's suppose there's like stars that are really far away. Some telescopes you can magnify it a lot, but it's just going to appear like a big blurry blob. Okay, that has low resolving power or low resolution. However, some what do you call it, telescopes? Okay, you can see they clear up, right? Instead of the blurriness, you see two small points, and so now you can see that, okay, those are two separate things. Those are two separate objects. That's called high resolving power. All right? So that's what resolving power is. You can see the examples here. On the left, low resolving power. On the right, high resolving power. So on the left, these two cells, they almost look kind of like they're the same cell. Right? On the right, you can kind of tell that, okay, those are two separate things. Does that kind of make
Yeah.
sense?
Yes.
Yeah.
Is-
I understand. All right. So now with light microscopes, there's a limit to the magnification that you can see with a light microscope. Right. So if you want to go beyond that, you have to use something called an electron microscope. Right. You understand that? So in the electron microscope, right, with the electron microscope,
what-
you can see all the way down to the level of an atom. All right.
Can you
So
actually
beyond
see atoms?
you can actually see atoms, right? So beyond a certain distance, beyond a certain magnification, the light microscope, the image is just going to be too blurry. You're not going to be able to resolve two things. So then you got to switch over to the electron microscope, all right? So usually, if you want to see a virus, you can't see it with a light microscope. So bacteria is right on the edge. You can see it, but you're not going to see any detail. There's not going to be almost any details. A virus, you pretty much can't really see it with a light microscope. Right? So these are the kinds of pictures that you see with the electron microscope, right? So
Oh
with the
my
electron microscope,
These are like 3D images, brother.
yes, you can see really amazing images. So remember, so what is this on the right? Anyone can tell me what is this on the right?
blood cells.
Blood cells, right?
No.
So you
Oh,
remember,
on
you
the
see,
right. Okay, no,
so you see the difference between the light microscope and the electron microscope, right?
Yes.
Look at how much look at how well you can see
Look-
all these red blood cells.
The details.
The detail is amazing, right? What is this
can
on the
literally
left?
see
Anyone know
every
what
They
this
look
is on the
like
left?
little pieces of
Viruses.
Smarties.
Virus cells or whatever.
Virus cells? Close.
Bacteria.
Not quite. So this is pollen, I think. I think this is pollen.
Oh,
Like,
really?
From plants. Yeah.
Oh,
So look at like the amazing geometry that's created here.
goddamn, brother. That's a lot of detail, brother.
And
There's a lot-
that's all
so-
in our body.
So you understand like if how Allah has created this. This is like who can engineer something like this amazing, right? Look how much detail it is. Right? So you can just sit here, all these pictures you can sit here and like study for hours, like how small and what is all this stuff here, right? Right. So
You can really get a lot of out of
with the electron
part.
microscope, right, it doesn't use light, it uses electrons. So it sends like a beam of electrons and shoots it at your specimen, right? In order
How
to
do they
view-
get electrons? Is it from like electricity?
Dude, electricity, man.
It
Okay.
produces electrons at the top. I don't remember. I don't know what exactly they're doing to produce it at the top, but you send a beam of electrons and it hits the specimen, and then you could look at it. Now the disadvantage is that you have to kill the specimen to see it. You cannot see like live things. So for example, in like with the light microscope, you could take some water from a pond and put it on there, and you can see like the little things moving around, right? You can see all the little protists and thing like moving around.
I think it like gives it like an electricity shot. It'll be like a lightning bolt for the electrons or whatever you're doing.
With the electron microscope, you cannot see living things. They have to be killed because you have to stain them with heavy metals first in order to see them. So let's take a look at what that looks like. So when you first of all, right, you produce these thin sections, right? To put it on a slide, it's going to be really thin. But what you have to do is you have to stain it with heavy metals, like gold.
Oh, so
silver,
that the electron actually go on. Okay, I see what's going on right here.
Right.
This is
So-
creating like a circuit, brother.
kind of like a circuit, right? So it has to be able to bounce off of these things.
Yeah,
right?
that's like the ground, brother.
So the heavy metal, right? Yeah, if you stain something, if you coat something
It
with
has to
heavy
be like
metal,
a conductor.
just a second. So if you
They
coat
probably
something,
use like copper.
wait a
right?
second. So
No,
I
man,
know,
hold
I
up.
know they use gold, but I don't know what else they use. I know gold is one. So they coat it, and then so but if you coat something with gold, right, now it's dead, right? That's not living anymore. So with this
Brother,
thing,
it literally says uranyl acetate and lead citrate, and then they wash it off. They put conductive tape on it,
right, so
and then they
They don't put it on it.
xylene on it, and then they put a heavy metal staining on it. That's a lot of stuff.
So
brother.
there's a lot of stuff going on here. So obviously the thing dies in the process, right? So you're not going to see any living things here. So that's the disadvantage.
Just kill it, brother. Who cares, brother? We need to see
Now,
this stuff.
Here we have two different types of electron microscopes. On the left, this is called scanning. On the right, we have something called transmission. What do you think is the difference between these two? How did they get these two different kinds of images? They have the same thing. They're both pollen.
Different view of perspectives.
Different perspectives, right? So tell me
That's
about that.
more of a 3D thing. And
Good.
the transmission electron is just like it's probably like transmitting through the
Mhm.
thing like the light microscope.
Okay, good. That's exactly right, right? So in the transmission, you're actually sending the beams through the slide, right? On the left, right, you're sending the beam at the specimen and is bouncing off of it. You understand the difference between like the dissection microscope and the compound microscope? You guys remember we talked about that difference, right? This is the same exact kind of idea. This one on the right, transmission electron microscope, right? You're sending the electron beams through the specimen.
Mhm.
So on the left, it's kind of like a dissecting microscope. You're just sending it's almost like you're shining a beam at the top, and then you're looking at the resulting picture. Right, Yusuf, you understand the difference?
Yes.
So this is the same thing, but we're looking at it in two different ways. Right? So this is
But
kind
this
of,
thing is 1,000.
so this is kind of showing the same thing, right? TEM, transmission electron microscope, right? And then SEM, scanning electron microscope. So you can think of it this way: the S stands for scanning, so it's kind of like scanning from the top. You're not passing it through; you're like scanning it from the top. Make sense? So with the transmission, right, you're sending the beam through the slide. With the scanning, you're like bouncing it off the top and looking at the surface. Right? So again, this is kind of showing you the same thing. This is a light microscope picture. This is a transmission where you're sending it through the slide. Here's scanning. You're just kind of looking at it from the top. Right? So transmission,
okay.
scanning. You understand the difference?
Mhm.
All right. So this is the transmission electron microscope. So again, these are some different pictures, right? So in these images, you're always going to see black and white. You're never going to get color pictures. You're never going to see it in color. So you might color it later with like a computer program and stuff like that, right? Later on, you might use a computer program and give it colors. So if you see electron microscope images with colors, that's something that they've done later on. That's not the actual picture that they got. With a light microscope, you can actually get the
So
color
no one
picture.
knows the colors of the atoms and like
right,
stuff.
right. So we don't have like colors of these things.
But isn't black a color?
Right. So these are some pictures. These are some pictures from scanning electron microscopes. Take a look at this, right?
Is that a mosquito or a fly?
Yeah, some kind of insect. I don't know what that is. So these are, and then these are some kind of small crystal, microscopic crystals, something like that. And these are from like a plant or something. I don't know what that is. Or maybe that's from an insect too. I don't know. Right. So these are also like pretty amazing pictures, right? Right. So these are just some kind of an overview, right? So at the top, right, we see these light microscope pictures. Light microscope. At the bottom, we see scanning electron microscope. All right. Can anyone tell me this one right here? You see this? Let's do like a guessing game. This top.
It
right
looks
one,
like a flower.
this one right here that I have my
Cross it.
mouse next to. What is this? Anyone can tell what this is? Yusuf, do you know what this is?
Where's but brother? Your cursor isn't even on the screen.
This,
So
this one right here. This one
blood
right
cells, brother. 100% blood cells.
here.
100%.
Blood cells. And what is all this like tiny like? This looks a little bit different, right? There's something different about this picture than before.
These are blood clot. No, they're not blood clots.
There's like lines too.
Yeah, there's lines, right? What else is there? There's some. What are those lines? Right. So it's blood and something else.
Brother, that's stuff that's
Bacteria?
eating the
Bac-
blood, brother.
Wait, what did you say, Anas?
Bacteria.
Bacteria. This is like a person who has a blood infection. Right? So in the
Oh.
blood, there's all those bacteria there. They're infected. They have.
Brother, that's what it looks like when you have an infection?
Yeah, that's what it-
Disgusting.
Yeah, so all that bacteria
Disgusting.
is there, right? What do you think
Ugh.
this one is here? What is this one right here?
Eh.
Anas, what do you think?
Brother, plant, brother. I don't care if
Yeah,
Anas
yeah,
gets
that's
it right
plant
or wrong.
cells.
It's plant.
Plant cells.
Yeah.
What about this one right here? Anyone know what this one is over here? Right, this is obviously from a scanning electron mic. I mean, transmission or scan. I think it's a scanning. No, I think this is transmission.
This is scan- this is transmission, brother. This is 100% transmission.
100%. All right, what is this one right here?
Because it's like
Mhm.
it's not 3D looking like
Right.
that kind of
Yeah.
scanning one.
Right. So what is this? What do you think this is?
I honestly don't know this
These are
one.
viruses. So each of these is an individual
really?
virus. Now you see that?
So they scam us on TV showing like these little smart
And then what is this one right here?
Stuff.
Final one. These two down here. What are these two ones right here?
monitor screens.
Monitor screen. It looks kind of like monitor screens. What is this?
It's like small.
Brother, that's a transmission, brother.
So these are atoms.
Transmission. For real?
Yep.
Huh?
Damn, those are small.
Damn.
So I think they use a
Those are
different technique. It's called like scanning tunneling or something like that. Scanning tunneling microscopes. So you can
That
see-
is really small, brother. You can't even see it, brother.
so
I can barely see that.
yeah. So these are individual atoms. So on the right, obviously they didn't.
They look like
Mhm.
they look like little LEDs or something.
Yeah.
Like really small
It's
LEDs.
like monitor pixels.
So remember, the one on the right is something that they colored. The scientists colored. The one on the left is the actual picture. And so they colored it to make it look like the one on the right. Right.
Mhm.
All right. So excellent work, guys. All right, let's go.